Good evening, everyone. Welcome to my garden shed. I hope it's going to be okay with internet wise, but you know what it's like in South London. We're terrible with electricity. So fingers crossed that this goes well. I have been studying flies now at the Natural History Museum about 20 years. I kind of still describe myself in the maggot stage as it is of my career. I definitely haven't pupated in something mature yet. But most of the time when we look at flies, we actually generally ignore the maggot stage because it's the adult one we use a lot for classification. And yet this is really bad because actually the maggot stage is arguably, or the larval stage is arguably the most important. With that caveat, though, I will say that a lot of this was very, very new to me and a lot of the different groups. And I realized that there's a whole lot more about the larval stage Diptera that we need to know, which we always knew that anyway. But there's a way more interesting things than I originally thought what was going on. So I'm very lucky. I do some. Here's me looking at mosquito larvae. And then obviously looking at dung pats and hoovering pigs, which is the usual thing. And we take this back to the museum and we look at them. And with the mosquitoes, we link rear. We have a huge understanding of what goes with the larvae. But our fascination with maggots has gone back millennia. And we've always been associated with maggots and death. And I've got this brilliant book called The Insect Transformations, and it's from the Library of Entertaining Knowledge, which, I mean, what better thing is that? And this was published in 1830, and it starts off, as all good books from the 1830s starts off with, it was universally believed that by the ancient philosophers that maggots, flies, and other insects were generated from putrefying substances. This opinion continues to be held by uninformed persons among ourselves. It's a real damning in a real way. But in fact, in many ways, we still don't know a lot about larvae and a lot about where they live and what they do. Popular knowledge, popular literature, popular opinions are all that these maggots. They are of a certain style and hang around certain things. I don't know what the book or a maggot is, which sounds quite good, but these basically should be ignored in favor of a lot of these books, which I've based a lot of my research on today, including obviously two of these, which I'm going to just recommend now. But it is Kenneth Smith's book on immature flies, which you can still get through the RES website. So that's very, very good. And it is amazing how much we haven't progressed when it comes to knowledge. So he writes about only 80,000 described species of Diptera, and we're up to about 165,000 to date. But within them, he said there's less than 2% have been described in the immature stages. Now, I heard figures that we're up to like 40% when it comes to UK Diptera, but this probably is maybe a little bit over-exaggerated. And there's still so much to know. And the families that we have concentrated so far are really the ones of medical or important economic importance. So a lot of the mosquitoes and or the crane flies or the tephritids and things like that. We're beginning now to look at some that are ecologically important. I'll talk about one of those in a minute. But most of the description for the immature stages has remained, as Ken Smith describes it, casual. Okay, and a lot of this is ad hoc, and it's really, actually, this is where the big pleas go out to everyone listening, is that you have to become your own maggot experts. So, for example, the chironomid, the non-biting midges, this is basically how I got involved with all things fly to start with anyway, because although I'd been looking at ecological entomology, I was working on a wetland, and my two supervisors, one looked at soil, one looked at trees. It wasn't very helpful for me, who played around in muddy puddles. And so they said, go forth and go to the Natural History Museum and speak to an expert there. And I was like, oh, really? There's experts at the Natural History Museum? And they let me behind the scenes, and that was their first mistake because I never really left. But I met a guy called Steve Brooks who explained about chironomids, and these are non-biting midges, and we know a lot of their larvae because of the bloodworms. And he said, Erica, these are really cool because they live in all these different environments. They do all sorts of different things, and they can live in different environments because they have many different types of their version of hemoglobin. In fact, they don't have them within blood cells that we do. They're just free floating around their bodies. And they have up to 11, 12, 13 different types of hemoglobin, depending on what species they are. And some of these are massive, massive molecules that are able to not only transport oxygen, but can store oxygen. And this in then turn enables them to live in these really dank and toxic, miserable lives. But we've been able to now look at these larvae and figure out the oxygen environment around them. And Steve Brooks, and we identify them we look at their teeth arrangement. So what looks like a crown, this is inside the pupal case of the head case, of the chironomid. These crowns, when they die, their body disintegrates, as we all will, but their head capsule is heavily sclerotized and that remains there. And we can go back then and we can dig up the mud from eons past, or soil cores or ice cores especially, and we can look what chironomids lived there, and we can develop what Steve Brooks described as a midgeometer. I love this term. And basically, they have been able to look at the assemblages of the last 15,000 years and work out which chironomids live in which oxygen environment. And so they're able to really accurately say what was going on. So not only can we now look at the past and we can say, okay, what was the oxygen events then? We can use it when we go forward in the future. So this is really important when we look at past European climate and future changes in climate. So these little chironomids and their ability to live in the most disgusting of environments are actually very helpful for us. And flies, full stop, are just amazing. Okay, it doesn't matter which part of the life cycle you're talking about, flies are amazing. And they're mega diverse, they're mega abundant, and they get everywhere. And this is also because they are able to separate their ecological niches between the larva and the adult stage. Baby bees stay at home feeding on adult bee stuff. Whereas baby flies, they are sent out to work from the beginning, and they have diversified what they feed on, where they live, all sorts of things ad infinitum. They've been able to disperse and conquer. So let's have a look at the life cycle of the fly. Here we go. Male fly, female fly, get together. It's all very quick. Sometimes it can be romantic. Sometimes gifts can be exchanged. Mostly not. And they do what all good flies do. They have some eggs. Some flies have many eggs. Some flies have hundreds of eggs. It all depends. And this egg stage is very important because this is when the egg starts developing. It's starting to grow in shape. We'll talk about the egg in a minute. And we move on to the larval stage. Often this is referred to as the maggot and then higher fly. And this is basically the feeding stage. And they go through a series of molts called instars. And the number of these, depending on what species, they vary a lot. But during each molt, the larvae sheds its skin. And within the lower flies, the old nematosferan flies, they shed their head capsule as well. So this is what is left behind that we can use to identify what's going on. And then they pupate. And this is probably, I think, to me, one of the most exciting things that you will ever learn about in biology. It is one of the most striking aspects of insect biology, where these insects undergo an almost complete and really complex process of reorganizing their body tissues. I just love the idea you've got to so far in life and then you could just reorganize yourself into different function. So most of the larvae organs are going to be replaced with adult tissues and adult organs. Those larval eyes, they get subsumed into adult eyes, all these different things. And the limbs for locomotion and the organs of reproduction are now in place. And here's a little video. This is, we have not been able to look in real time. Is this working yet? Of what's been going on within the pupal case, but we have loads of different advances now. And one of those is micro CT scanning. So we're able in real time to have a look at what's going on. And this is a pupa. This is you can see the gas bubble, and the gas bubble gradually shrinks as the larval form disappears and moves in to get the adult form. And if I jump to various bits, here we go, you can see the head developing. And gradually, we're seeing the features develop more and more. So it's amazing now, and this is really important when I talk about this later for forensic entomology and things like that. We can have real hour-to-hour understanding of the development process, what's going on in the pupa. And it's not continuous. You get stops and starts, and we're beginning to understand it. So it's great. We're at a period of time now where we can utilize technology to help us understand some of those missing questions. And then when it comes to getting out of the pupal case, here we go. This is, and if you haven't spent half an hour in a garden watching a fly emerge, I mean, this one, bless it, it's a parasitoid. So this is a little bit funkier because this is emerging from a bee. But you can see here, this is a canopid, and it is thick-headed flies, and it's blowing up its head. It's one of the higher flies. It has this teleneal sac. It blows up an airbag to burst out of its puparium, which is absolutely amazing. Not all of them are that dramatic, but quite a lot are. And if you want to know what's going on with how they then go for the next stage, because not only are they bursting out of they're blowing up their heads, they're hardening the exoskeleton, they are expanding their wings, so they're taking in lots of air to blow up the wings. And we're only still really beginning to understand not how the process is, but how these wings fold out and what's going on there. And there's a new publication here on wing expansions of Drosophila. Drosophila is our model organism. It's the only model you should ever pay any attention to. And this is in a this is a preprint looking at wing expansion. It's a very interesting read of what's going on there and how all the different crinkles will iron out as it goes on. So let's go back into the egg stage. So this is a generalized egg of a Diptera or all insects, and it's composed of the chorion. This is the tegument, the outside, the embryo, and its accessories, along with the nutrient reserves, the yolk. Now, most of them are rich in yolk. There are, because obviously this is flies, there's always exceptions, and the bad boys and girls in these exceptions are the tachinids. Okay, many of those do not have any yolk. There is much cleaving going on in early development, and we'd often think about the egg stage not being very, very defenseless. Okay, that's what the eggs were out there. There's nothing they can do. But we're realizing more and more now is that they have some defense mechanisms, and they will develop this cellular layer outside, and they are able to set up an immune response to bacteria. So we have in the chorion, you'll have some differentiation. You have some erypiles, which are these holes within the egg, and it allows gas to diffuse across the surface of the eggs. But this obviously lets other things get in, but they're very active to make sure that if bacteria come in with it, they try and get rid of it. So it's not just this kind of weird static, well, externally static phase. There's a lot of activity apart from development going on. So here's some of the eggs. Obviously, you should all have Alan Stubbs's Stubbsy to me, crane fly book. This is great. And at the beginning of it, in his latest book, his chapter on the early stages, he states, very little has been published on the eggs, and it's easy to gain the impression that they are rather featureless. It's a little bit harsh, but for example, many of the cranefly eggs are mostly elongate and oval. So here's a good example here with the, this is a Holorosia. This is not a UK genus. Okay, we can see them there, but there are some funky stuff starting to happen, even just within the eggs, even within the dipulids. And I love this. This is a lovely paper, which included so it was Graham and Jeff and various other people, obviously on a summer holiday, looking at crane flies. And they had an opportunity to catch one of the tipulids here, Tipula escuriventris. And they reared out, they kept her long enough to rear out some of the eggs. And you can see here, they've got this coiled thread at the apex. So this is external of the egg. And this is believed to actually be able to, when they were placed in moss and whatever, it kind of acts as like an anchor to kind of wrap around this vegetation so these eggs just don't drift off. So that's quite funky. The chironomids, the non-biting midges, these eggs are laid in the most amazing gelatinous masses. I've come across many of these in ponds. I kind of like think of them as like seafood slugs because they have that kind of like weird texture. And the eggs are arranged in rows, they're folded into loops, they're embedded, and there can be within each egg mass, I mean, some, you can have maybe up to 1,000 eggs. So the mothers were obviously assuming that there's going to be a high attrition rate. I mean, I imagine they'd be quite tasty for any aquatic vertebrate swimming around trying to eat them. And they are laid on the water surface, in the surface, under the water, in emerging vegetation, etc. We've got the biting midges, the ceratobigonids. Again, these can often be found in gelatinous substrates, egg masses, sorry, but these are also terrestrial. So a lot of them we don't quite know yet because they're in very many different habitats. But we are beginning to look at the ultrastructure of these eggs. And we're beginning to understand more the phylogenies of all these families based on the eggs. So many of the adults will have very similar morphological features, but we're now starting to go back and try and find the eggs or rear these adults, produce the eggs to understand how they relate to different species. So it's really good at finding sibling species. Mosquito larvae. This is where I spend most of my time with mosquito eggs. So mosquitoes split into two subfamilies. You've got the culicines and the anophelines. And I like to describe the anophelines, they're the photo at the bottom, as hot dogs in their bun. You can see them like sausages wearing, I don't know, rubber rings. This is what they look like. And anopheles, they lay between about 50 to 200 eggs, single eggs at a time. And they've got these little floats float around the surface. Whereas the culicines, which is that raft you'll see on your left, they kind of look like a load of bowling pins stuck together. And she will lay them in clusters in these rafts, and they kind of support themselves in this way, floating on top of the surface. And they may contain up to about 300 eggs. We've got the Aedes, which is a type of culicine, and they kind of like a little bit haphazard. But if you see, you've got loads of sculpturing going on the eggs here. Arguably some of the best egg sculptures, though, belong with the tabanids, the horseflies. These are amazing. The genus Tabanus and Hypermitra, two very common genera in the UK, they can make multiple layers. And honestly, I couldn't get any photos without copyright. That was an issue. But they look like pyramids where you've got layer upon layer upon layer of these eggs together. And they can lay up to 500 eggs in one go. Now, what I hadn't realized till a couple of weeks ago as well was that there's one species of Tabanus, not here, it's found in North America, that exhibits parental care over its egg mass. So here you've got, this is a Goniops chrysocoma, and the female, she will actually stay with her eggs until they've hatched. This is rather sweet for a mother. I gotta love that. We've got some other eggs, canopids. Now, these are parasitoids. Parasitoids are always a bit of fun. And these eggs require the development of a hymenopteran host, bees, wasps, etc. Now, a lot of them, the female will actually use her egg laying tube to actually slice open the back of a hymenopteran, oh dear, and insert her eggs in. But there's a genus called Stylogaster, which I'm going to say they've weaponized their ovipositor. And what she does is she's able to like aim and fire. So her eggs are like little like bullets that she fires off in different directions. And what they do is they hang around all the big army ants. And so when the army ants, and I've just seen these in Costa Rica, when they're on the move, the mass of them, they push up all these other insects who definitely don't want to get eaten by the army ants. And it scares a lot of them out of their home. And the stylogaster is like boom, boom, aiming. And if you have a look on this little image here, you can see on the eyes of all these flies and on the thorax and the abdomen everywhere where these eggs have penetrated. The idea of having a larvae crawl for your eyeball, it's amazing. Move on to the syrphids, the hoverflies, and we can really start to see some amazing sculpturing going on here. This is a really lovely image of a microdon, and this is really quite useful taxonomically because the microdon, the three species, the Myrmicea, oh, I've lost the other two. Yeah, three species adult that they weren't able to properly differentiate very well. And so they went back and looked at the larvae and then they looked at the egg stage. And so this paper I've given you here is able to show all the differences within the eggs. But also, as you can see, it really looks in and you can see the eggs are trapping small droplets of water in these processes. So they're keeping themselves nice and moist because eggs do quite like a bit of it. One of the eulids, the pitcher wings, and we have been looking at SEM work. So the scanning electron microscopes looking at the external coverings of these eggs. And you can start to see all the beautiful differentiation going on. Here's some more. So we've got some pollinia that's going on here. We've got some psyllids, spila going on here. It's all this. So it's really beautiful, different ways we can differentiate between all of that. And all of these are very useful because obviously they're letting exchange of oxygen in and out. The mussids, the houseflies, I thought three different types of mussids for you to have a little look at. So this is the Hematobia irritans. Okay, now this is the hornfly, and she oviposits or lays about 78 average eggs. I love the fact that's the average during her lifespan of like 6 to 7 days as an adult. But so she can lay many more, but that's the average it can go. And these are laid in fresh manure, and there's a lovely description that often even if a cow hasn't finished copulating, defecating, she's in there, which I imagine is quite messy at times if she gets that a bit wrong. We got the stable flies, Stomoxys calcitrans. Any of you have hung around stables or cow sheds, you've met some of the adults, and they are rather affectionate towards a lot of humans, often very painful. And the female's here, and because she's getting a blood meal as well, she lays up to 800 eggs. But she will lay them in clutches, but she needs a separate blood meal between each clutch. So hence why she's so needy in her attention towards us mammals. These get laid in, eggs get laid here in feces, decaying or damp organic material, etc. And then you've got these. This is Musca autumnalis, a really common housefly. And these are lovely because not only than the others, they have this respiratory horn. These little things, these protuberances coming off. Now, we see these structures across many families of flies, so sepsis, Drosophila, dung flies, house flies, etc. And these enable the eggs to extract oxygen when they're in really wet, damp, aquatic conditions. So they're able to actively get oxygen in these very, very liquid environments. So you can imagine, because and a very, very wet feces or something like that, because these lot love fresh cow dung, being able to get oxygen in that environment is quite important. Then tachinids. Now, tachinids are pretty much game on for everything. Indirect, direct, mixed oviposition. And these are just fantastic and do everything, but I will point you all in the direction of Chris Ray Tachinid group, because they've got lots of information. And actually, I should point this out full stop, that all of you should join the Diptera forum, sign up. It's an amazing organization, and there's a wealth of information ready available for lots of people who've got more clue about everything than I do. So please go forth. So within the chinids, you've got indirect oviposition. So these eggs are just depositive around. Some of them will be next to the host. And some, the eggs, because the adults go through, well, I'll talk about their what they do in a minute. But some of them will basically lay eggs that hatch immediately and the larvae are off. So the larvae, some will just sit there and hope for the best and wait, while others are active trying to find their host, which is quite cool. But you then have some that are direct oviposition. And Matt, these are images where I haven't acknowledged them. These are all for Matt Bertone, an amazing photographer. And I just think, and he commented on this, the idea of sitting there knowing that your death is just sitting on your head and there's nothing you can do, and you just think that's the worst place for an egg. And the females will use many olfactory cues to locate the habitat where the hosts of her larvae will be found. So it is quite fantastic. But not only will they lay them on or near, but some of them have the most amazing ovipositor. Here we go. This is not, I think the first time I saw this, I don't know whether I was both horrified, amazed, or I was just, look at that for an ovipositor. The ability of movement in that. So a lot of the tachinids would try and do this. Poor caterpillars, but oh well. And then you've got the slightly lazy ones, or maybe the very clever ones. These are the botflies. Now, the human botfly, sadly, we don't get in the UK. One day I'll get one and it'll all be cool. But what they do, because the botflies, I don't know if you ever come across them, a lot of them are quite cumbersome and quite noisy. And the human botfly has realized that actually to get to us and some of the larger vertebrates, it uses something else. And they use this intermediate host, like mosquitoes, horseflies, houseflies, that do all the dangerous stuff, like going up to the humans. And it is only when the mosquito, for example, is feeding off to the eggs suddenly hatch, the larvae then drop through. So here you go. This is in the collection at the NHM. This is one of the mosquitoes. And you can see these 4 eggs actually stuck on the side of a mosquito. The evolutionary process of how that happened in the first place, genuinely don't know. And then my favorite eggs aren't even eggs at all. So in the hippobosids, in fact, the whole of the hippobossoidea, which include the hippobosids, these are the bird flies and the bat flies and the tsetse, they give birth to live young. So the larvae are nourished by this internal milk gland, and it produces these milk proteins, these endosymbiotic bacteria, and it feeds them. And so she, bless her, because sometimes they give birth to something that is a mass larger than themselves, which as a female, it just makes me feel very, very uncomfortable. They contain enough yolk to develop into larval. I'm just going to read you a quick bit from the little insect transformation. So this is Remer, and he was the one who really started looking at these flies, and he gives a lively narrative of his discovery and the solicitude of his servants to find him females ready to deposit what he at first took for eggs. He was so anxious to hatch these supposed eggs that he carried them in his pocket by day and took them to bed with him at night. How cute is that? So I'm not saying that you should take maggots to bed or flies to bed to look at what's going on, but I think that's dedication to the cause. And this is an interesting point because actually flies have many different ways of laying eggs or larvae. So oviparity is a traditional way, what many of the insects do. You just leave an egg, hatches outside the body. That is the norm. But then you get this oviparity, okay, where the female retains the eggs inside. They are nourished by the egg itself. Um, but the eggs, they can either hatch pre leaving the body or just after. So really, really precocious. The larvae, the eggs are just basically there. Poof, you've got a larvae. And this is really important because this is really useful when you've got ephemeral food sources. So things that are going to disappear, you want to be right in there straight away feeding. And then going back to the hippobosids, you've got this viviparity. So this is development of embryo in the mother and is looked after by the mother. So she properly is pregnant, which is quite fun. Now all of these different ways, all of this ovoviviparity, etc., flies love it. Here's an example, 22 families of flies across Diptera. And we're talking everywhere from cecidomyids all the way up. So it's just this plasticity and how they have the eggs, how they larvae, also emphasizes massively the variety of form in their feeding habits and where they live. There we go. So let's get on to larvae proper. Oh gosh, I'm going to have to race through this. The larvae is the feeding stage. Okay, lots of feeding going on, and they feed on pretty much everything. I do find it very lazy when everyone goes, oh, it's just on the feces and the dead food and whatever. No, you name it, they do feed on it. And it's really important because this is where most of our environment is cleaned up because of them. Where are they found? Pretty much everywhere. I luckily was in California in January. Thanks, taxpayers. Very much appreciated. Luckily, it was the American ones. And this is me giggling because I'm now collecting some maggots that live in the tar pits, the La Brea tar pits. So these larvae are feeding on all the dead material that has fallen into the tar pits, can't survive. These larvae can. It's amazing. So what makes a dipteran larvae dipteran? Well, theoretically, all of them, their ancestral trait is that they have no true thoracic legs. So we talk about them as precocious. They developed out the egg earlier, so they haven't got the 3 pairs of legs that you see in Coleoptera, Lepidoptera, all of the other groups. But be careful, they're close relatives, the fleas, found in Antlion amorpha, they too don't have thoracic legs. In fact, I've got the flea collection at work, and it's amazing how many similarities between the pair of them. But they do have a lot of proto-legs. So they have these podites. Look at these cuties. And again, these pro-legs, these fake legs, we see all the way through the mussids, all the way through the Diptera, sorry, from Eudixids all the way upwards. Sadly for us, we don't get these in the UK. These are just amazing. These are the mountain midges, the Deuteroflebidae, and these are arguably the sister to all Diptera. So they're in with the fly, maybe they're not. And these occur in fast-flowing streams. And they're little prolegs, they're little suckers, and they just enable these little larvae to walk around the bottom of these streams. Now, we do get some in UK. So we got some of the Pediciidae, the hairy-eyed crane flies. This is in the genus Dichronota. Okay, and they've got these nice fleshy prolegs on 5 of their tergites. So it's quite cool. As you can see that they've got these retractile heads. They're quite weird. And then for even more obvious ones, in the Atheriaceridae, look at this. Look at Aphrex. It just looks like a beast walking around with these amazing sucking prolegs as they just suck around. It's great. How do the larvae breathe? Well, again, no fly larvae. It's like they just went, no, let's complicate it. And you've got all these different breathing systems. So we have the many spiracles, A, B, and C, versus the few spiracles, D, E, F, versus the no spiracles of G. So, and a lot of this variety goes on within the lower flies, what we used to call the nematocerans. We get this complete hodgepodge of what's going on with breathing. So for example, this lower, the holomneustic, okay, we've got spiracles all over their body. And this is found in some of the bibionids or all of the bibionids and genus the Pachnura, which exists this. And this is an interesting thing, actually, what I'd like to point out is that within families, you can have this variety of form as well. And we will talk about this later on because this just the spread of diversity when it comes to their morphology is just phenomenal within Diptera. So we got all these different breathing, these spiracles. And then the mouth parts vary as well. So a lot of them, a lot of the very primitive larvae, or the primitive flies, sorry. So the lower Diptera, they have often chewing mouth parts. So they are organized, the work horizontal. So they kind of meet in the middle. It's a bit like, clapping hands going on. So you've got this slicing and shredding. A lot of them are filter feeders and things like that. And then gradually, as you go up through the higher flies, so into the Brachycerans, we got a reorganization of jaws to they're almost with the higher flies, like pointing down as fangs. And at this point, they're kind of scraping. So when you think of like these things that are consuming flesh and how they scrape off the flesh, marvelous. I've got myself quite hungry now. And we also have a change in head capsule across the larvae. So you've got the lower flies, the nematoceran flies. So this is, you can see a phantom midge here with its amazing prehensile antennae. I'll talk about in a minute. These have got these very developed head cases. We talk head capsules. These get left behind as they go through each molt. The mosquitoes, really nice sclerotized head capsules. Annoying for anyone who ever studies mosquitoes. When you look at their larval skins and pupal skins, or the arrangement of all these bristles is taxonomically important. That's a lot of fun. But as you move up through the nematocerans and into the Brachycerans, you get this reduction in head capsule. So here's a tabanid, a horsefly, and these are what we call hemicephalus. They've got a little bit of a head left, but it's not much until we get up to maggot sensu strictu. This is where we're happy to call them the maggots, and they've basically got no head form whatsoever, and they have this cephalorangeal skeleton. Okay, and this is amazing. It comes out and does that. And it is the morphology of that skeleton, along with their spiracles, is how we identify them. But their form is more and more simple as we go up through higher flies. And this has been problematic when it comes for identification. So let's romp through the lower nematocerans. Okay, the lower Diptera. Here we go. We got some of the crane flies. These are the leather jackets. All sorts of fun. And they're all sorts of fun because look at that. Nothing makes me more pleasure than looking at the spiracles and the appendages of the spiracles of cranefly larvae. They do look like Tamagotchi larvae. They do have a purpose because they actually do try and protect the spiracles. Why they're such form, who knows? But honestly, do yourself a favor. If you haven't done it already, go into your garden, find some cranefly larvae and look at their bums. It is pure pleasure. And again, lots of easy identification of cranefly larvae going on here. One of the associated family, the short-pout craneflies, I just love these. There's about 227 species of these in the UK. This is one of the genera. This is Hexatoma, and that turnip-like apparatus is its abdominal termites. And now this one, these live in sand, in riverbeds, banks, etc. And I presume this is basically them weighting themselves so that they don't kind of float off this enormous turnip-like structure. We move on to the cylindrotomids. These are the damsel craneflies, and there's only 4 species in the UK, but they are the most specialized, some of the most specialized nematocerans when it comes to their larvae and what they're doing. Really nice to identify. Lots have been done, and we can separate them into the 2 that are, here you go, the cylindrotomini, which and the cylindrotoma and the Dogma, they're terrestrial, and the other 2 are aquatic, and they vary their form slightly depending on their environment. Trichosarids, these are the winter gnats, 12 species in the UK. They can't retract their head into their thoracic segments. And so that's, although they're superficially like the crane flies, that's a nice little way of being able to have a look at them. What I love about these ones, they breed everywhere. So dead and decaying plant material, fungal material, cow dung, carrion, and distillery waste, which is very pleasant. But these things, because of their ability to live in some really very liquid and very disgusting environment, these have turned up in Antarctica, in sewage stations. And it's like, wow, that's quite amazing. Obviously, they're trying to eradicate them there because it's not seen as very good. But wow, that's an explorer, isn't it? Ceratophragonids, the biting midges. Oh, I love these. I got quite sidetracked by these because they live in both terrestrial and aquatic environments. They do all sorts of things. But a lot of them, and you can see in Matt's photo here, is that they have this secretory setae on their head and their body, and they produce these hygroscopic antibacterial substances. And this is quite amazing, being able to do that. So, don't eat me. And apparently they can repel ants and things like that by these substances. I got really sidetracked at this point, and I was looking up all different sorts of. These are in the genus Forsythia, and Forsythia, we know the adults because the adults are the pollinators of chocolate. These are ones found in Australia. Look at those beautiful balloons that they walk around in. These are amazing little fluffy things. This is another one found in, again, Tasmania. And this one looks like it's like, hussars. They're little like with these little fluffy pom-poms going around. And to detract from the bird, if you come along and eat me, you can just eat this fluffy pom-pom. Great. I just genuinely would love to see one of those. Sadly, our ones aren't quite as exciting. Mosquito larvae. Okay, these are tumblers. These are lovely. I don't know. There's a beautiful descriptions of playing with these, and I spend many a time tapping the surface. And they split within the two subfamilies. The Culicines have got this big breathing tubercle, where the Anophelines don't. They lie flat underneath the surface. There's a third, like these are within the Culicines. This is a Toxirhynchites mosquito. And these are amazing. They're, we used to think they're their own family. We've sunk them back in the Culicines. A bit, but these are highly predatory on other mosquitoes. And it's really upsetting if you're a naive mosquito worker out in South Africa and you've got one mosquito species to find, and you go out and you get all this larvae and they all look the same until you come back and one of them is grown enormously and you're like, oh, that's odd. And then you come back and they've eaten all the other mosquito larvae. These are huge. They're highly, they just consume everything. They're amazing. And we're using them for biological control because the adults do not blood feed. The females don't at all. We got the phantom midges, and these are amazing. Because look at them, they look beautiful, but they also have these prehensile antennae. So they can actually go and capture their prey. So they can get other things like mosquito larvae, and they even get some Daphnia. So they're kind of like a crustacean. I know, it's nice, isn't it? And they're able to crush or impale their prey. It's brilliant terms, and bring it into their larval mouth. And what you can see here, these little circular structures, these four structures on their thorax, these are their air bags. So they enable them to kind of float around quite nicely. They're absolutely, if you see them, they're completely see-through. Till sadly they die and then they go completely opaque. It's quite amazing. Across, there's 2 out of 3 families are not found in the UK, but what I wanted to show you as we begin to talk about is this respiratory siphon, which is attached to a spiracle. So this we find all across diptera larvae, all different families have it, and it enables a lot of the larvae to live in really disgusting environments, but still breathe air from the surface. So we can see it. These are in, say, for example, the UK species Cuptrids. Okay, so they have this one long, massive, partially or fully retractable siphon, which is quite amazing. The owl midges. these are quite interesting in that they exist in all sorts of environments. You often come across them in your sinks or in your compost. So, and they're quite cute again. Some of them will also, like the little anopheles, will have little rubber rings to kind of hold them up on the surface, which is quite cute. Window midges, again, what is quite interesting and what I brought out these because within the gnatty groups of flies, you often have very clear, very obvious white long bodies, very simple, but with very defined head capsules. You can see on this one here, the images, you can see the spiracles, okay? So it only has the 2 sets of spiracles along the body, so that's very easy to see. And then you've got scatopsids, the worst common name ever, the minute black scavenger flies. Now, these larvae, although looking very cute, upset me because these are some of the most important pests of oyster mushrooms. So this is one of the most important economic fungi in the world, and these lot are desperately just trying to consume as much as possible. They can do secondary damage, so not only is them consuming it by causing, they can introduce pathogens, mites, and nematodes. And this is a problem with a lot of the herbivorous or fungivorous. It's not actually them consuming it per se, it's actually what they can transmit. Bibionids, brought this little photo up because actually this is where very mindful of misidentifications, because the bibionid, the march flies, their larvae superficially look very much like the tipulid. So they're very often very dull grey. Some of them will have orange spines, but then most of them are soil dwellers, and they can get huge densities. And cultivated land as well, they can be massive problems to crops, but they're just beautiful. Gall midges. This is where common names are really bad because not all of the midges are gall midges. Accessotomides. They do many things. In fact, many are free living. So here we got some that are brightly coloured red and orange and pink and sometimes white. And all of these have this distinct sternal spatula. And this little bit here on the little larvae. Now, this enables them to get out. It helps them to emerge from their pupal case, okay? It's able how so they can get out and quickly move on. And it's best developed in the soil-dwelling larva, where it's used for tunneling. Sorry, it's used for, so the larvae are using it, not getting out of the pupal case, sorry. It's used for tunneling to build a pupal cavity, okay? But they do say that although it's quite distinct amongst the different species, it gets utilized a lot, so it suffers wear and tear. So older larvae are just a little bit more battered. But a lot of the cestomides are gallers, okay? So I've got to rush through all of these. So here's some more of the different ones. And herbivorous plant insects are divided into the gills of the chewers, the suckers, the miners, the drillers, and the gall makers. And for galls, check out the British Plant Society, and also check out the sites, nature spots and things like here. They're great for it. Here's another type of galler. This is within the Tephridids. But what I wanted to highlight with the cestomides were not the galls, but these really weird larvae. So these are genetic larvae. So these larvae are sexually mature before reaching adulthood, which is quite cool. So the larvae have little larvae in them. They produce these larvae, which then eat them from inside out, which is, I just can't even mentally think about what that must be. And they can do this for several generations. And then they can suddenly revert to the normal form and go through pupate, and then the adults can form. What they don't have is they don't have that appendage for scraping because they don't need to. So they are literally just going through it. But when they don't develop the sterna. So, but this reappears when they are going to go through pupation. So they switch it back on when they're going to become like the normal form. I also wanted to highlight with the sestamized, the leaping. Okay, so a lot of boinging movement. So it's not just your traditional walking. A lot of these insects will move by leaping, and there's numerous families that can jump. Look at that. Whee. Here's another family. This is Longcaeids. And you can see how it uses its mouth parts to grab on and before it boings off. It's great, isn't it? Okay. And look, this is how as an example, is a lovely paper here, came out in 2022, and it shows you all the different families that do this leaping behavior. So the ones we know, the classic ones are the pyophilids, the cheese skippers. It's kind of in the name, but we can see it exhibited by lots and lots of different flies. The dark-winged fungus gnats, the skyarids, live in a large range, and they move along with their creeping welts, which I'll talk to you in a minute. But you also, and I'm very pleased to say that we get these now in the UK, you get these ones moving en masse. So these are long columns of what they call armyworms. Is that going to focus properly? Oh, I think it's a little bit fuzzy, sorry. But they're absolutely phenomenal. Just if you can watch a video of armyworms just marching along as they're moving along, all moving to try and look scary to protect them. I think they look rather cute. The fungus gnats proper. So these are when we start getting to the really economically important ones. What I love about fungus gnats, not only the adults, is the ability to a lot of the individuals of the UK to actually find out more about them. And one of them I am going to name, her name is Judy Webb. And within her kitchen, she rears up a load of different fungus gnats from different fungi. Now, this is the sort of dedication we need to understand more about our fauna in the UK. So I'd like you to all go home and go away and whatever, and to start rearing up your own fungus gnats and things like that, because host records are something that we really need. Now, closely related to species, the Cheroplatids, these are the fungus gnats as well. They've got a very broad general, but a lot of these we're getting into the predator ones, whereas all the Mycetophilids just ate fungus. These ones are starting to eat other things. And 3 of their genera are bioluminescent or have bioluminescent species within them. So we've got Cheroplatis, one of those is found in the UK. Ophelia is another genus. But the big one is the Arachnicampa. And these are about 5 species, these found in New Zealand and Australia. These are fantastic. And the bioluminescence is created in their Malpighian tubules. It's their version of the kidneys, and produces this unique, obviously it's unique, bioluminescence enzymes that combine, and they lure in their prey. So they have these balls, these strips, and the little moths are like attracted to all the beautiful light and go, ah, and then hit them. And it has this lysing agent, this necrotic agent that dissolves the insect, and they feast. So they're like really deadly fisher. And what I love about them is they're a beauty spot. So everyone is going to these caves going, isn't it amazing? Isn't it beautiful? No, it's death and carnage. But yes, go ahead and celebrate them. Quickly moving on to Brachycera, because I've got, I don't know, about 5 minutes to do all the rest of the flies. And I will highlight one of the recording schemes of the UK, the soldier flies and their allies. Lots of amazing information. So here we've got the old flies. Just going to talk about these because these are one of the few flies I know are named because of their larvae. Because their larvae, these head capsules, are the needle that is meant to be used in leather work. And so that is why these things have been called all flies, after the all needle. So here we go. Here's an example of why the soldier flies and their recording scheme is so good. We've got all these amazing identification within the Stratiomyids, the soldier flies. Now, the Stratiomyids, they just like doing everything. Aquatic larvae, terrestrial larvae, semi-aquatic larvae, the 48 species found in the UK, just everywhere. And they have these different appendages associated with them. But the one I'll quickly talk about is this one, which is now on a UK species list, Anatia lucens. This is the black soldier fly. And this is the one which will arguably save the planet, honest. So this is what we're actually using to rear up, and we're using it for food. So we've been eating insects in our diet for thousands of years, and about 2,000 insects are eaten globally. Only 2% of the insects eaten are flies. Weirdly, people don't like eating maggots, don't know. But they have looked at this, and this little soldier fly is it's a hipster, and it really is organic recycling. It does all that. And it itself is 40% fat, 40% protein, 30% fat. And it's able to really rapidly consume waste. So the idea is that we get rid of a lot of our waste and we rear up these larvae and then consume these larvae. Now, we're not doing it as maggots per se, but we're actually grinding these up to make as flour. So we can actually use them in other products to make it much more sustainable. Horseflies, these are great. These are some of where I really get excited, because these and some of the other ones we've already mentioned are part of the neglected venomics groups. So these horseflies are badass predators, full stop. I love it. There's a beautiful description of a BBC explorer researcher out in America, and he was looking at this frog, and this frog was kind of suspended in the sun, and he tried to pull it, and there was something pulling against him, so he pulled and pulled and pulled, and then out popped, when he pulled it, was a little larvae, and the larvae was one of these horseflies. And these horseflies, I mean, how many times have you got a larvae actually consuming a vertebrate like that? Amazing. You do not mix with these larvae. They live in all sorts of environments. Some of them can create little mud nests. Some of them, so you get some semi-aquatic ones. And at work, we've actually got these larval tubes created out of mud, which is quite amazing. But talking of things created out of tube, these are Vermilionidae. We don't get these in the UK. Really annoying, because these are worm lions, like the ant lions, and they live in these little larval pits. They're great, and they are vicious. And as the little ant comes wiggling along and then falls into the pit, they just reach up there with a raptorial front end and just stuff them. What's quite cool as well in these pits, and with actual ant lions as well, there's also some horsefly larvae that live in there and feed off the waste as well. It's a good habitat, go for it. And talking of apex predators, we've got the acilids, the robber flies. Now, these generally long white larvae prefer drier habitats, sandier soils, very predaceous. But some of them will live in decaying wood. This is my only attempt at a decent image of Larvae flava up in Scotland. And it was a pupal case, so it's slightly cheating, but you can see its head appendages to show you how it lives in this decomposing wood environment. And here's where we start fumbling a lot, because we don't know a lot about larvae. We've got a lot to learn about them. This is for Ovids. We don't know. We can't describe any of the larvae, really. They're very active. We've just had some recently brought into the museum. We're going to sequence. We think they're all vicious predators, thanks to based on their mouth parts, but really hard to buy apart. And so very small differences in their head capsule, therefore poorly described. So 8 out of the 14 UK species, we have some, but if you look in Alan Stubbs' Soldier Flies and Their Allies book, it's very poor. Not having a go at Alan. Acrocerates. Now, within Acrocerates and bombylids, parasitism gets ramped up massively. So these larvae are fantastic because they go through 2 different life forms. You have the first one, and this is where you get the I can't even remember how to say it now. The planidium, and this is the first stage. It's the active stage. So this is the one that is after its host. Now, in Acrocerids, its host a spider, so pretty game on. But in bombylids, this can be anything from bees to orthopteroids to all sorts of things, depending where on the planet, and Lepidoptera, where they are. And what we've got here is the second stage. So we have that really active planidium, the first stage, and then you've gone from the toddler to the teenager, because now this is the stage, just stays put, it's found its host, and it just is going to consume it. Whether it's endo or ecto depends on the species. For example, this is some of Matt's images of a little, look at that, little larvae. And this is an ectoparasite on a Zalicopa, so the carpenter bee. And he's been watching it develop, and they can develop to huge beasts. I mean, look at that. I don't know what it'd be like to be consumed like that, but seeing something feeding on you growing like that bit must be quite demoralizing. We move through the empids. Oh, look, we've got our little prolegs going on again. Again, these can be aquatic, semi-aquatic. And then we've got hypotids. And this is really quite cool. So this is one of the species you get in the UK. She's got viviparous larvae. So she just basically squirts fully formed young larvae all over dung, which they develop. What a way to be a mother. Some of the dollies, again, the larvae are not well known. We think they're very predaceous. They live in many places, and these are quite exciting because a lot of these families are now like on seashores. So they're eating all sorts of things on beaches, which is definitely worth going and have a look at. Right, now we move on to the higher flies. Okay, and these where we get also, this is where we're beginning to get the maggots. We get this reduction in head capsules, we get everything. And I love this. Steve Marshall in his book on flies, which you should all get anyway, and take a day off and just read it. It's beautiful images. But when he talks about these sort of flies, his description is brilliant. There's a lot of M's. Most maggots macerate various kinds of muck with their mouth hooks. Brilliant, from Marshall. And this is what's going on. They're macerating with their mouth parts. I've got to thank Jane, who's actually in on this talk, for sending me these images. So this is in the Eisgeiser. Okay, the platypesids, the flat-footed flies, and these are great, these flies. Some of them hang around bonfires. There's a genus that do. I have no idea where they live, but a lot of them love fungi. And look at this, decomposing honey fungi. It looks absolutely disgusting, and the larvae love it. So she describes how when she went around, she saw the females laying the eggs on the fruiting bodies about 1 to 2 weeks previously. And this is the development when she's come back. And they pupate in the soil surrounding this decaying fungus, and they will overwinter there to emerge the following autumn. And she just said that there were several thousand larvae in one clump. How amazing. And then I have to thank Jane again, because I have this on my desk, which I think is amazing. Now, this platypesid, this Acathamia wankowitzi, which is a silly name, this is the only invertebrate in Britain known to cause a gall on this bracket fungus. So bracket fungus is a really hard. Okay, this is just old, but it's still how they were. And these are amazing. And they have these like colonial, like loads and loads of them living together in this amazing thing. Syrphids, the hoverflies. These have taken that respiratory siphon and run with it. So they have a telescopic organ. So these are within the Aristolani subfamily of the hoverflies. Aristolus tenax is a classic one, but Mythraber florea, all of these ones, they have these beautiful things. And they can actually, so often the telescopic, the telescoping siphon is like 1 centimeter, but they can expand it to 5 centimeters. So these are extreme bog snorkelers. They are going from it in these environments, and they're really good at getting in and recycling and converting this into useful protein again, i.e. themselves. But there's also the other ones. Within the Syrphini, this is really cool, because these are the Ophidophagus ones, and they're just going for it. So these now have turned themselves into biological warfare units. So each little larvae can consume between 600 to 1,200 aphids before it grows up. And some really lovely work by Will Hawkes and various other people looking at migratory hoverflies. Turns out, if the migrants that meet up with the local population when they get together produces much more fecundity. We do like the immigrants, as it were. And these are coming in, and she will lay loads more eggs, and it's brilliant. So we get these migrants doing all sorts of amazing things of pollinating on the way over, dying, leaving these nitrogen packages, i.e. their dead bodies as they go, and then they're producing these exceptionally important aphidophagous. So these really are your gardener's friends. And then you've got some other ones within the surface. So this is a microdon. Oh, absolutely amazing. And these are very different from many of those. The peculiar shape of them made them, when they first got described, Van Hayden in 1823, he thought they were a slug. I mean, yes, maybe. And so there's been a little bit of taxonomic confusion with them. Their larvae feed on ant larvae, which is great. They're going in there and just munching away under the eyes of the ants. The horrid phorids. Now, these are quite interesting because along with the surfids and the phorids, we're starting to, although we have got it in a lot of the lower Diptera, talking about the miasis. So these are any flies that cause an infestation in humans. And there's been some amazing look at all these different things. And we've been called into hospitals, for example, if they've got an outbreak of phorids. And these phorids are they're most ecologically diverse, and they can get anywhere. And I mean anywhere. And these have been found in humans in the urogenitory tract. That's how they can live absolutely anywhere. So they seem to have developed the most hardcore method of living out. And finally, guys, is the little long copterid. Because look at these. So these little larvae, to me, they just look like little wood lice. Okay, they're about 4 millimeters long. They've got these long, hairy bristles. They're found in really similar habitats, under logs, dead leaves, etc., vegetable general matter. And also, these things get themselves into vegetables. They get themselves in between the leaves of Brussels sprouts and sweet turnips, things like that. And so they often get consumed by us by mistake. Oh. Right, into Scytophora. So these is very diverse lineage, right? We're totally maggoting now, right? We've got a lot of these now are in really soupy feed like liquids. And so these are using this kind of pharyngeal filter to concentrate the microorganisms. So you can see this, the oral ridges, the CR on these little diagrams. So these are just concentrating. And a lot of these move, by the best term ever, by creeping welts. Okay, so this is one of the Tephritids, the bedfly. And it just highlights where all these creeping welts are on, and that's how they move along. They've done loads of amazing robotics looking at how maggots move, because they're actually trying to do some highly non-invasive surgery and using the idea of a maggot to get around our body system because of the flexibility of the maggot and the way it can move, which is amazing. So here we go. Here's some. Look, picture wings, eulids. They are about 20 of them. These are, this is the problem now. We're getting into things that are economically important. So a lot of these are found in our crops, in our vegetables. And these affect our onions and garlic a lot, which I think is a little sad, being such a lover. And here this is some of the leaf miners. And I'm going to highlight, this is Barry Warrington. Now you need to check out his Agromyces site, because look at this video. And you can see this little larvae. These are between the leaves, between the upper and lower of the leaf, the surface of them. And it's just feeding away. And look, you can really see how the pharyngeal hook is just reaching in and just shredding away as it's going. He's got some amazing images and some amazing videos. Now, many of the Agromyzids are host-specific, but some of them are very polyphagous. They can have many hosts. So there's all sorts of information, all sorts of knowledge we still need to know. Here we go, here's some more. A lot of identification we can do on their mines, the shape of their mines. So you get these linear mines, or you can get some nice blotched mines. He's got a load of images associated with those. And these are another group that some of them are miners. Okay, these are mainly grass flies, mainly on grasses. I think you can see why, but they can be major pests of cereals. However, one of them I think is quite funky and probably disgusting to the average person, but I love them because they're parasites. Their larvae live under the skin of frogs. Look at that. And you can see, if you look really carefully at these larvae, look at it. You can see its little anal spiracles going. The breathing tubes at the back of them in the second photo. They're apparently, some people are like, they're not really detrimental to the frogs. I don't know if I totally believe that. As this lady says, but they don't live here, sadly. These are from Australia. Now, all the Syrphids got all the credit for the Ophidophagus when it comes to Diptera, but there's some unsung heroes as well, and these are the actual aphid flies, the Chamaemomids. And there's about 185, 3 species everywhere, 32 in the UK. And these are really important predators of aphids. Yep, and they do all sorts of things. And again, quite easy to identify, so we should learn more about them. But how we do know a lot about everything is because of the Drosophilids. So this is the model organism we talked about before. And these do everything. Again, they've got that linear form, that quite white, opaque form that many of the larvae do. But this is the one that's turned up in the UK and causing a lot of concern. I love it. This is the very first world problem here because it's attacking our soft fruits. However, it has been known to attack grapes. And at that point, I start paying a lot of attention because if they touch my wine, even I might not like them. So Drosophila suzuki, they can have significant effects. And it, like looking in the States alone, pest damage in 2008 was 500 million. So they really do have quite an exciting time. Diphydrids, these brine flies, little bit of attention to diphydrids. Those little flies that I was collecting in the tar pits were the diphydrids. The little flies that live in petroleum pits, diphydrids. The ones that live in carrion, diphydrids. The ones that can eat the inside out of spiders, diphydrids. The ones that live in brine, diphydrids. It's like diphydrids are like, go for it. They are amazing explorers, amazing adaptations going on. There's 153 species in the UK. Go for it. Go and find these amazing creatures. Don't worry, we're nearly there. We're on to the beautiful botflies. We have to go out on a big high. I love a botfly. This is the larvae of what we call the subdermal botflies. This is the Dermatobia hominis, the human botfly. It's not just on us, but we're so us-centric. This actually was from my friend's head. I'd like to know that's now in the collection, which is quite exciting. If any of you get botfly, please donate it. It'd be really good. And this is the one. This is on a Phaniid, which is quite exciting because it's like, well, why is it on a Phaniid? Because that doesn't actually come to us. It hangs around our latrines, etc. And obviously, the oostrader's got a bit confused. And look, these larvae are pretty much ready to go. We caught this in a baited meat trap where this Phaniid was caught, and it's like, oh. It's like bittersweet for these larvae. But look at that. How many larvae are on its abdomen? Genuinely was one of the most amazing things I've ever seen. Now, I haven't had one yet, but this guy, you've got to look up, Gil Wizen, read his blog about him rearing his own botfly. Maybe not do it before dinner, but it is truly exciting. The second group of botflies are the gastroenterhinal. You get some mammoth botflies here. Look at those larvae. Absolute chunks of those. And it's quite sad because actually these are some of the most endangered species on the planet because a lot of these, look, this one is found in a rhino. And now we're looking after huge amounts of conservation associated with rhinos. Actually, we're getting rid of this fly. So we're not giving the fly the same value as we are giving to the mammal, which I think is ridiculous. And then my favourite botflies are the snot bots. And these live in the pharynx, in the nasal cavity of many a mammoth, mammals, etc. This is the classic removed deer head dissection, which just is like extreme. And this is one of the reindeer botfly heads. If you're a reindeer and you're that close to a botfly, It's pretty much game over for your nostril. She will squirt up to 200 larvae into the nostril. And my favorite scientific name ever is Cephalopena titillator, which is the camel nasal botfly. And so once the larvae are once they're matured and ready to leave her nose, I've got to stop doing this on my nose, they agitate the nose and she just snots out hundreds of larvae. Amazing. I'll do a little bit on the root maggot flies, because this is, I think, a quite good example of things that when they go through their life cycle are very different. So when we think about these, we think of them as being really important agricultural pests. However, their adults are massively important pollinators. Massively important pollinators. And again, Will Hawke's work has just showed how many of these are migrating around, coming in here and pollinating. So it's one of these things where we've got to be really careful that we don't think about it linearly. We've got to think about how they change. Also, these we can get some gastrointestinal issues with these because we swallow them a lot because they're in our food. Quite exciting. Again, with these are faniids. Look at those, very cute, but I'll move on. And the stomoxycalcitrons. So again, this is where we want to talk about myiasis. And I'll quickly move on from this because I'm mindful of time until I actually do that. The sarcophagids, the flesh flies, Dan's got a really good recording scheme associated with these. These are absolutely opportunistic with their larvae. And these are oviviviparous. And so they were basically eggs ready to go, or larvae get squirted out immediately. And many of them are necrophagous, but some of them will parasitize on other arthropods. So here's one, it's going to go inside and eat a millipede, which is quite exciting. That's not a UK species, sadly. But let's give the final word to the maggots that everybody hates. Everyone goes, oh, they're really disgusting. This is terrible. The blue bottles and the blowflies. Now, these maggots, bless them, I get such a hard press, and you need to go to Olga's recording scheme to have a look at these. But these are some of the most important for several reasons. We've utilized this ability for them to go into these dead and decaying bodies for our own good. So the first one is forensic entomology. And we've now been studying forensic entomology for 1,000 years. So there's your classic little image, there's your caraphorid. And we have been using their larvae to age bodies. We've been using their larvae to understand all sorts of things when it comes to crime scenes. So this is really useful for understanding the dead, but also it's really good for helping the living. So now on the NHS, you can have debridement therapy. So this is great. You can get your own maggots eating away at your necrotic flesh. And now this isn't, apparently we really don't like maggots close to us. So nowadays they come in what looks like little tea bags. And if you have a wound and it may be ulcerating, you have all these nasty things going on, these little maggots will go in and clean up that wound amazingly. And not only do they clean up the wound, they release enzymes that stimulate your flesh to grow, and also they can get rid of bacteria. So things like MRSA, they eat. So these little maggots are superheroes. So vastly over time, I'd like to say it's a complete romp through the flies. I didn't even get onto the pupal stage, but please do check out all these recording schemes associated with the UK Diptera. Join the Diptera's forum. It's very friendly, it's very lovely, and lots of help. And I just say this is only the start. There's a whole lot of pupa to go next and what's going on. So thanks, Matt, Soldier Flies, Diptera's forum, everyone else, and especially thank the maggots. Right. I will stop talking.
Thank you so much, Erica. That was absolutely fascinating. Those who have been lucky enough to be on the call tonight have really been treated. And you know, I found it was hard not to I just wanted to pause you and just go off and read. You know, I could just read, you know, many hours of then just come back and carry on. But it was, yeah, it was difficult like that. It was so interesting. All this, you know, it was so well illustrated with amazing photos and videos, and you just wants to just should just go and indulge it for
I,
the
I
next few days.
genuinely, I'm as much as I've cursed you, I have to thank you because I was just going down little maggot high roads, being more and more amazed by the different things. And you know, all this stage has to do is feed. That's all it has to do. And yet the different ways it's gone about it is like, this is amazing. Absolutely incredible how much diversity exists.
Yeah, there is so much for people to go off and explore from your talk. I mean, yeah, really, really grateful for all your time and all the research that you put into that. Must have been
Yeah. Might have to write a book now.
yeah, I think you should. So I don't think we should. Yeah, I think. Yeah, I mean, yeah, I don't think we should have a full-on question and answer session now, having, given how long you've given us of all that amazing information. We had someone asking about providing links to all of the articles. I think a lot of them, you had a lot of scientific papers there that are not going to be open access anyway. But I mean, there was lots of websites links that you gave and
Yeah.
the
I mean,
recording.
I was hoping that a lot of it, if you're recording it and they can access it, because I wanted to credit as many people as possible. And if there are papers that people are having problems, just to email, and I should hopefully be able to provide them with a PDF of it. But yeah, that is the problem with a lot of hidden behind all of these.
But there was you did give so many links on your slides there that people are going to have really plenty to be going and looking up for themselves. And I've made a long list, but I kind of gave up. um,
Sorry.
it was just too much.
There's a
No, it's
lot of
fantastic.
information.
Yeah, there was. So I think, yeah, I think it's 5 to 9, and I've really enjoyed that. I'm sure everybody else has. So I think yeah, I think we'll leave it there, really, for this evening, unless you're.
I mean, I don't mind on it generally because I went so far over. If you want maybe 5 to 10 minutes, it's up to you. Or is everyone absolutely shattered? I may have shattered everyone. I'm so sorry, everyone. I may have caused your brains to turn into a little maggot mush. Yeah.
Well, I could give a couple of questions. If someone had a really
Yeah.
pressing question, then
It's got to be a really good question.
it's got to be, yeah, it's got to be your best question. So, and in fact, if you do have that question, you can just unmute yourself and speak it if you like. I don't think I can see anything in the chat. Or you can just email Erica afterwards.
I think maybe that's probably going to be easier. I think I have brain fried everyone. Sorry, folks.
Not not a question, but one quick thing. My brain has turned to mush. It's been absolutely fabulous, but I am now going to need a glass of wine to recuperate.
Me
But
too.
people were talking about papers behind paywalls. In many British libraries, there is something called access to research. You can only access it within a library, but it gives free access to a lot of articles that are behind paywalls. So if there's any of these you want to chase up, it might be worth a trip to your local library.
Oh, that's a good plug for local libraries as well. Well done.
Yeah, that's great. Thank you. Thank you very much, Paul. Okay, well, yeah, I think we'll leave it there then. And yeah, just extremely grateful, Erica. That was brilliant. That was really great. And yeah, I hope we'll have to try and get you back for a continuation in
Puppy. But we must bring wine next time. Thank you. Thank you very much, everyone. Thank you.
Okay.
Cheers.
Good night. Thank you very much, everybody.
Thank you. Cheers, everyone.
Everyone, bye.